Okay, let's give people a minute to log in. We pause that, start recording, and start the share. I had to stop the share because I can't hit the recording button. At least I don't know how to hit the recording button in this screen, but when I'm sharing, but when I'm not sharing, I know how to do it. All right, I've already asked if there are any questions. So let's begin. Sorry, I'm not seeing my icons. We're going to be finishing chapter 2 today and hopefully getting started with chapter 3. On Thursday, we'll be finishing chapter 3. And then also note this week, tomorrow, at 11:59 p.m., you got to take the first formal quiz. If you haven't downloaded the Respondus Lockdown browser, please do that. There was one student this term who had trouble, and I haven't heard anything further, so I assume they contacted the IT department, and the IT department got it working on their system. So download it and make sure it's working, and then take the quiz. You can take it from 11:59 p.m. tonight till 11:59 p.m. on Wednesday night. Any questions about that? There are 2 parts. There's a essay questions and short answer questions that you need to take. You do not need the Lockdown browser for that. Many of the questions are, I wouldn't say there's a wrong answer. There's no one correct answer, I'll word it that way. I suppose there are several incorrect answers, but there's no one correct answer. So you just have to give a good answer, and you can look in your book, you can talk together with other students. However, if you get together with other students, remember, if you use your study buddy's answer, you have to put it in quotes, or else it's plagiarizing. And I haven't had that often happen, but last term I had some students turning in the same answer. And especially when it's wrong, it's quite obvious to me that that was the same answer and that they were sharing their answer and they just turned it in, and so they got 0 on that. So don't plagiarize, all right? The second part of the quiz, quiz 1B, is a timed exam. I don't remember how long you get, but it'll tell you when you do it. You get 1 minute for each question, and then something like, well, a few extra minutes for the entire quiz. So it is a timed quiz, and it's timed for the purpose. You simply can't look up every answer. You might have enough time to look up 1 answer. If you're really quick, you might even have enough time to look up 2 answers, but you won't be able to look up any more. So this is a timed exam. The intention is so that you don't get to look up the answers. Any question about that? And those are multiple choice, the Respondus Lockdown browser, timed exam. The other one, it is timed in the sense you have only 24 hours and it has to be finished by Wednesday at 11:59 p.m. But you can take as long or as little time you want to answer those questions, assuming you get it done within 24 hours. All right.
Will this be covering chapter 1, 2, and 3, or?
No, it says up here, that's a good question. It says up here what it's covering. Both of them are only covering chapters 1 and 10, and the start of chapter 2, meaning what we covered in week 2 only, and labs 0, 1, and 2. So lab modules 0, 1, and 2. The quiz will be on those topics. Okay? And if you don't know exactly where we ended in chapter 2, just Gosh, let me think about that now. Just go about halfway in chapter 2, okay? And that should be more than enough. All right, any questions about what we're doing?
I have a question about lab. Is it Tuesday or Thursday that we have lab?
Oh, whenever we only have one lab in a week, I will be, I don't know, going through it on Tuesday and then staying there for the majority of the time on Tuesday. On Thursday, we will meet, but that's all we're going to do. I'll say, are there any questions? And I will be there for about 15 minutes, unless there's lots of questions. And then if there's no questions, I will end. And you guys, if you don't have any questions, you don't have to show up on Thursday. Okay? But I will be there for 15 minutes on Thursday to answer any questions. And you have to get the lab done by Saturday. Okay? And I'll talk a little bit more about that. The lab has to be done on Saturday or it's late. A number of students, I guess I'll talk about it now. A number of students have not been turning it in on Saturday. And it is true that I'll tell you which are wrong and then allow you to have more time to do it and put in an update. But it is due on Saturday, and if you don't get it in on Saturday, I'm going to be considering it late. Just to let you know. I'm not sure I'll make that applied for this one, but we will see. All future ones will be, because a number of students are not turning it in on Saturday. All right. Let's end that and get back to our lesson. So we had talked about DNA, and we discussed this slide here. Let me get rid of that. Are there any questions about DNA or anything on chapter 2 so far? All right, so we discussed DNA is double-stranded, composed of the sugar deoxyribose. It has 4 nucleotide bases, which are the steps on the ladder, and the sugar phosphate bases. I don't have a good picture of that, but what's holding the strand together are the sugar phosphate. So right here is the sugar phosphate, and then a base, and then a sugar phosphate. So the DNA is the backbone of the, the sugar phosphates are the backbone of the DNA molecule. And the bases, all they do are connect the two strands of DNA together by hydrogen bonds. Now let's move on to RNA. What's the sugar for RNA? Is it deoxyribose?
No, it's ribonucleic acid.
Yes, it's ribose. It's ribonucleic acid for RNA. The sugar is just ribose. It has a phosphate, ribose, and then a nitrogen-containing base. And there are 4 bases. We mentioned that 3 of them are the same, the A, the G, the C. The T is replaced by the base uracil, or U. Once again, the sugar phosphate connect the RNA strand, and it's the backbone of the RNA strand. Is the RNA single-stranded or double-stranded?
Single-stranded.
The RNA is single-stranded. There are 3 major kinds of RNA. Transfer RNA, or tRNA, messenger RNA, or mRNA, and ribosomal RNA, or rRNA. Now, the majority of the genes when we look at the DNA, code for a protein. So when those genes are transcribed, what are they going to be transcribed as? So the majority of the genes, when they're transcribed as RNA, what are they going to be transcribed as? Okay, are they going to be transcribed as tRNA?
Messenger?
Yes, they're going to be transcribed as messenger RNA. So when does a gene, when is it transcribed as tRNA or rRNA? You got to remember what I just said. The majority of the genes code for a protein, so they are transcribed as messenger RNA. The other genes code for either tRNA or rRNA. And when those genes are transcribed, if they code for tRNA, they'll be transcribed as tRNA. If they code for rRNA, they'll be transcribed as rRNA. So a gene will be transcribed as one of these three, depending on what the gene codes for. Everybody got that?
Yes.
Yeah.
Okay. All right. Another part of nucleic acid we need to talk about is adenosine triphosphate. I mean, we could talk about all the, what do you call them, the nucleotides, but adenosine triphosphate is the primary energy source for cellular activities. That means when a cell needs energy, for whatever reason, it tends to get the energy from ATP. ATP has ribose, of course, and adenine, that's why it's ATP, and then 3 phosphates, why it's triphosphate. And when the cell needs energy, it breaks this first or the third phosphate group here, because where there's a squiggly line, there is a high energy bond. And when that bond is broken, it releases a lot of energy. And the cell can use that energy for its energy needs. If the cell needs more energy, it can also break this bond, the second phosphate, because it's a high energy bond as well. Any questions about that? So ATP is called the energy currency of cells. Currency is something we use to buy things. It used to be used on the old days all the time. Obviously now you can use credit and debit cards, but it's still handling money in a sense, meaning the bank or the credit card company is handling the money, but and then you pay it later, or if it's coming out of your account, you pay it right away. But the point is that cells, when they need energy, they tend to take it from ATP. So ATP is called the energy currency of cells. Any question about that? ATP is made by dehydration synthesis. It's broken, the ATP, excuse me, right there, the ATP right there, by hydrolysis. You add water and then break off that phosphate group, releasing energy, and the energy can be used by the cell for its energy needs. And that results in the water being joined to the phosphate and the ADP, adenosine diphosphate. And then the phosphate comes off and it's inorganic. Phosphate is always inorganic. You can make ATP by taking ADP, adenosine diphosphate, and adding a phosphate group. And it takes a lot of energy, but you need to add the energy. And then this reaction can go this way, forming water and adenosine triphosphate. And that's the dehydration synthesis reaction. This way, and then hydrolysis is that way. All right, so what is adenosine triphosphate? What is it used for in the cell?
Currents, or well, energy usage and kind of, it's like the currency of energy, basically.
Yeah. All chemical reactions, meaning all big biological molecules, ATP, DNA, RNA, even starch, all of them have energy, which, if you break it down, you can release the energy. And our cells can break down glucose to generate energy, and we'll talk about that later. And what happens is wherever there's a chemical bond, there is energy in that chemical bond. But particularly our cells aren't good at harvesting energy from chemical bonds. Our cells typically get the energy from ATP, and our cells can get it from glucose, but there's really only two reactions that we can do to break down glucose to utilize the energy from glucose. And most of that is done in the mitochondria, and we'll talk a little bit about that later. All right, any questions about that? Hopefully I didn't confuse you, but I'm just saying that wherever you have a chemical bond, let's go over here. Like this first phosphate could be broken off and that would release energy, but our cells don't do that. We're not good at breaking this bond as well as harvesting the energy. And we could even break off, if we were really good, break off the oxygen here and that would release energy. But our cells don't do that at all, okay? We do not break off the oxygen group from the phosphate to release energy for our energy needs. Let's just word it that way, because I imagine in a chemical reaction, like if we were using phosphate in a chemical reaction and changing the phosphate molecule, we might break off that oxygen. I'm not positive if that happens or not. To my knowledge, it doesn't happen, but it's possible it does. But we don't do that to release energy and then use that energy for our energy needs. And we might not even do it at all. It might have been clear there. Okay. We can burn sugar, but actually most of that is done in the mitochondria for obtaining energy for our energy needs. Sorry, I'm thinking here. Like I said, most of it is done. It is true with glucose, you can undergo glycolysis in all of your cells. It doesn't have to be in the mitochondria, but that doesn't generate much energy for our energy needs. Most of it, the energy in glucose is converted into the energy needs in the mitochondria. Hopefully I haven't confused anyone. All right, let's move on. Let's talk a little bit about hydrogen bonds. What molecules have we discussed in Chapter 2 that have hydrogen bonds, giving the molecule important properties?
We talked about like water, amino acids, some proteins, nucleotides, all those have hydrogen bonds, don't they?
Yeah, it's right here. Nucleotides, nucleic acids. Most molecules do have hydrogen bonds in them. Most biological molecules. The exception is lipids. Lipids are nonpolar, so they do not form hydrogen bonds. But there is one lipid that does form hydrogen bonds. What lipid is that?
Is it
Phospholipid?
phospholipid?
Phospholipids, yes, all right. And what would happen to these molecules that have hydrogen bonds if the hydrogen bonds were to suddenly disappear?
They wouldn't function properly.
They wouldn't function properly, is certainly correct. The protein would unfold and denature, and then it wouldn't function. What would happen to liquid water if all the hydrogen bonds were to disappear?
Just like evaporate. Wouldn't
Yeah,
it?
it would be gaseous water. And then DNA, if all the hydrogen bonds were to disappear, your DNA would no longer be double-stranded. Okay? All right, any questions about Chapter 2? If not, let's begin Chapter 3. Okay, like normal, I usually have an introductory slide giving you the name of the topic, Chapter 3, Observing Microorganisms, and giving you the major goals of this lesson, not the entire goals, just the major ones, and a rough outline of what we're going to cover. So you need to know the terms: resolution, refraction, stains, basic and acetic dyes, smear preparation, what's a mordant, and peptidoglycan. You also have to understand the sizes of organisms and the properties of microscopes. And we will briefly look at the sizes of large organisms, but mostly we're going to talk about the sizes of microorganisms. Understand the major differences between the light microscope and the electron microscope. So you're going to need to understand the sizes of organism in relationship to the microscope, and then understand the major differences between a light microscope and an electron microscope. And by the way, somebody in one of the labs answered the question about what kind of a microscope do we have in the lab, and I was shocked. Somebody actually said an electron microscope. And I can tell you, in no microbiology lab I've ever been in had there been an electron microscope in that lab. All right? Only special laboratories have an electron microscope, and generally they just have one. Usually a big college will have one electron microscope. I don't think Clark College has an electron microscope, but a big university, usually they have one, like OSU, Oregon State University, Washington State University, they have one. Understand while staining improves resolution. You should know that. We will discuss it. Know the 3 different types of stains. There are simple stains, differential stains, and special stains. And then for the differential stain, understand the Gram stain and its procedure, and the basis for its differential staining. All right, any questions about what we're going to cover? If not, let's move on. When we're discussing microorganisms, you need to know the units of measurement that are used in science, especially in microbiology, and it is the metric system. You are expected to know the metric system on quizzes and exams. We're not going to talk about it a lot here, but in one of the labs there's I think there's some quizzes, which are not extra credit, they're optional quizzes that you can take. And there's some optional activities, and we'll go through it a little bit, both in the lecture and in the lab. You should know all of these terms here. A millimeter is a meter is 1,000 millimeters. 1 millimeter is 1,000 micrometers. You don't need to know that way, but you should know. Let's blow this up a little bit so you can see it better. You should know, oops, too far. You should know that 1 kilometer is 1,000 meters. 1 meter is the base unit for the length. You don't need to know the decimeter, but you should know the centimeter. And a centimeter is 1/100th of a meter. You should know a millimeter, which is 1/1000th of a meter. And you should know a micrometer, which is 1/10⁻⁶ meters, or 1/1,000,000th of a meter. And you should probably know a nanometer, which is 1/10⁹, or 1/1000,000th of a meter. And why you should know the micrometer in particular is that's the size of things we view under a light microscope. The average size of a prokaryotic cell is around 1 to 3 micrometers. They can be smaller, I'm talking about the average size, and they can be bigger. You should know the nanometer because that's about as small as you can see under the electron microscope. I'm talking about a normal electron microscope. But of course, an electron microscope can view a cell which is around 1 millimeters. And I'll show you a graph of that in a minute. And you should know that the metric unit is based on tens. So, for example, well, let's start here. I'm not going to start there because I don't know what 10 to the 5th is called. I'll have to look it up. But 10 millimeters equals 1 centimeter. 10 centimeters equal 1 decimeter. And like I said, you don't need to know that term, but it's useful to know. And then 10 decimeters equal 1 meter. I used to know this. 10 meters equals a decameter. You don't need to know a decameter. 10 decameters equal 1 hectometer, which you don't need to know that term either. And then 10 hectometers equal 1 kilometer. Or 1,000 meters equal 1 kilometer. So you should know this chart here from 1 nanometer up to 1 kilometer. You should get that memorized if you don't know it, and what the conversions are if you don't know how to convert them. And then you should also know that you can just switch out a gram here, and then you can call it a kilogram, a decigram, a centigram, or you can call it, instead of a meter or gram, you can call it a liter. And then there's a kiloliter, a deciliter, a centiliter, and then everyone should be familiar with a milliliter. That's the unit we use in the lab. You do not need to know how to convert from the metric system to the English, and English to the metric system. Like the only thing I remember is 2.54 centimeters equal 1 inch. And that conversion is difficult. Okay, and you don't need to know that conversion. But you should know the metric conversions, and you should know the metric system. Any question about any of that? And why do we need to know the metric system? Besides that there will be exam questions.
It's just a much more accurate way of depicting sizes.
That is true, and that's why we use it in science and medicine. And because it's used in science and medicine, you need to know it. Because in this class, it's a science class with a medical bent to it. So you should do that. And then when you're out in the real world, you will be using the metric system, assuming you become in the medical profession. And it is true that when we weigh somebody, we don't use the metric system, although oftentimes they will record it in metric and then take it in English. But like when you're giving somebody a shot, it's usually measured in milliliters or microliters, something like that. And oh well, we'll just word it there and leave it there. Another reason for using the metric system is shown in this chart here. This chart here. Here we're looking at the countries in the world that do not use the metric system. What do you notice about the countries in the world that do not use the metric system?
What if you want to?
There aren't very many. Most of the countries in the world use the metric system. There's only 3 that do not, and the United States is the only big one. Okay? And actually, we are in the process of switching over to using the metric system. Thomas Jefferson, whenever he was president, passed a law or whatever that the United States switched to using the metric system. And as you can see, we have been very, very slow about converting. So we are in the process, maybe in another 100 years or so, we might finally switch. You should know the sizes of things. And this is just telling you a typical size of some microorganisms. And this is sort of a range. And obviously you can have eukaryotic cells smaller than this and eukaryotic cells larger than this. Does anybody know a eukaryotic cell larger than this? Everybody should. Everybody's seen a eukaryotic cell larger than this. What is it? What's the largest cell you've seen? Somebody can go in online. You should also know about the size of typical microorganisms, and I have no idea why they use this range, because this is close to the size of an average prokaryotic cell, but then this is about as big as a prokaryotic cell can possibly be. Most prokaryotic cells, their average size is between 1 and 3 micrometers. They can be smaller than that. They can be down to 0.4 micrometers. So 0.4 micrometers would be the smallest. And then close to 10 micrometers is the largest prokaryotic cell. Nobody went online to find out what the biggest eukaryotic cell is that you can see.
An ostrich egg?
I see.
Yeah, well, not many people have seen an ostrich yolk to the cell. It's not the entire egg, it's the yolk of the egg. That's the largest cell. Not many of you have seen an ostrich yolk, but a lot of you have seen, actually all of you have seen a chicken yolk. Okay, when you crack open a chicken egg and you see the yolk there, that's the largest cell that most of you have seen. Has anyone ever seen a turkey egg or cracked open a turkey egg?
I have not.
Nobody has. Okay, I'm sure none of you have cracked open an ostrich egg, so that's why I didn't. And most of you haven't seen an ostrich egg. I've never seen a real ostrich egg. Okay, but that would be the largest cell. The yolk of an ostrich egg is the largest cell on the planet. But all of you have seen the yolk of a chicken egg, and that would be the largest cell you've ever seen. As I mentioned, prokaryotic cells can be as small as the largest virus. If we're talking about the virus diameter, the very largest virus, the diameter is 0.44 micrometers, the smallest diameter being 0.02 micrometers. And with viruses, you can talk about them by their diameter and by their length. The largest virus by its length is the Ebola virus, and it is 1 micrometer in length, but it is not the largest virus in diameter. Megavirus is. And the reason is Ebola is long and skinny, and so its diameter is actually smaller than megavirus. So megavirus is the largest virus by diameter, and megavirus is as large as the smallest prokaryotic cells. And in this slide here, let's look at it, the slideshow. Looking at the size of things, you should know the ranking. Like a human is bigger than a chicken egg. Chicken egg is bigger than a frog egg. A frog egg is bigger than a eukaryotic cell, like a human egg cell. Eukaryotic cell is bigger than a bacteria cell, and bacteria are bigger than viruses. Viruses are bigger than large, like molecules, like proteins, or a prion isn't a molecule. That's well, it might be a molecule. We'll talk about that later in the term. Anyways, viruses are bigger than proteins, and then large molecules are bigger than small molecules like water, which are bigger than atoms. So you should know the ranking. You should know that a light microscope can see the smallest bacteria. Is that true? Close enough. And can see things up to about the size of a frog egg. Obviously, you're not going to see the entire egg under the microscope, but the microscope can be used to look at frog eggs. The electron microscope can almost get down to seeing atoms. It definitely can see small molecules, and can, a regular electron microscope, can see eukaryotic cells. Like I said, you do not need to know the units here. You should just know the ranking. And note that there's a mistake here, and I put a correction right there. So let's talk a little bit about microscopes. We've had this in the lab. We use in the lab the visible light microscope, and we use a compound microscope. A compound microscope is a microscope that has 2 or more magnifying lenses. And so there's one lens, the ocular lens, which magnifies. And in our lab, we only have 10x magnification in the ocular lens. But you can have a different magnification here. I've never heard of a different ocular magnification than 10x other than the 20x. But you could have anything between 1 and 20x for an ocular lens. Any question about that? I suppose it's possible to have above that, but I've never heard of it. And then the second magnification lens is the objective lens. Typically, the objective lenses are 4x, 10x, 40x, and then 100x, with the 100x being the oil immersion lens. And then the total magnification is the ocular lens times the power of the objective lens. You should know the different parts illustrated here, and we'll talk a little bit about that better here. The ocular lens, also called the eyepiece, you should know that. You should kind of know the tube. More importantly, know the arm. That's what holds the microscope together and what you carry the microscope. One of the things you carry the microscope with. You should know the objective lens, the stage clip, which is not shown too well. You should know the condenser and the iris diaphragm. On this microscope, the iris diaphragm is underneath the condenser. Sometimes the iris diaphragm is above the condenser. You should know the illuminator, which is also called the light source or the lamp. The coarse focusing knob, that's the big one. And then the fine focusing knob, that's the little one. You should also know the rheostat, which in this case I assume the rheostat and the switch to turn it on and off are the same, meaning you move the rheostat to turn it on, and then you move it further to turn on the light further. Any questions about the parts of a microscope? If it's mentioned here, you should know it. If it's mentioned in the lab, you should know it. Alright, this slide is showing you a microscope that is not a compound microscope, and it's a simple microscope. We talked about it a little bit when we talked about Antony van Leeuwenhoek. And this is the start of the in-class activity. So you'll be working on this for your worksheet for lecture 3. And note this week you have 2 worksheets which are due. The worksheet for chapter 2 and the worksheet for chapter 3. So make sure you get both of those worksheets due by Saturday, 11:59 p.m. on Saturday. Otherwise, they will be late. All right, we're not going to talk about these because you're covering them in the in-class activity. And in the in-class activity, you're looking at the electron microscope and how it differs from the light microscope, meaning it uses electrons. I'm not going to examine you on the different types of electron microscopes, but there are the 2 types discussed here, the transmission electron microscope and the scanning electron microscope. And then there's a 3rd type, which the slide is discussing, but I'm not going to question you on this, meaning you're not going to be tested on the 3rd type or the 4th type, the atomic force microscope. That's an electron microscope. It gets down to seeing very small images, objects, whatever you want to call them. I'll briefly discuss this slide. Which cells are easier to see? The cells on the left or the cells on the right?
Cells on the right.
Cells on the right. The only difference between these cells and these cells is that these cells have been stained. And that's the purpose of staining, is to increase the contrast between the cells and the background of the slide. Whereas this cell here has very little contrast between the cell and the background. And so if we stain a cell, it makes them much easier to view. And that's the purpose of staining cells. There's one more slide I want to talk about. And that is, we're looking at two different stains. A basic stain, which stains the cell itself, and either does not stain the background. In this case, the background is very lightly stained. And that's a basic stain. And then the other stain is a negative stain. It stains the background, but not the cell. And both of them allow you to see the cells because the stain is increasing the contrast between the background and the cell. Any questions about that? All right, so this is the last slide of the in-class activity. In this slide, we're talking about one of the important differential stains, and it's the most commonly used differential stain in microbiology. It's the Gram stain. Another commonly used differential stain used in microbiology is the acid-fast stain. It's a differential stain to determine or see if the cell has mycolic acid in it. And if they do have mycolic acid in it, it retains the acid stain. One of the stains is acid, and so we call those cells acid-fast. Any question about any of that? All right, so the differential stain is the most commonly used differential stain in microbiology, and one of the most common, if not the most common, stain used in microbiology. It was invented by Dr. Gram, Hans Christian Gram, in 1884. And he was initially using it to be able to determine whether a patient's cells were infected or the patient's tissue was infected from a bacteria. And what he developed the stain for was that the patient's cells would stain gram-negatively, meaning they'd be pink, and then the bacterial infection would stain purple or gram-positively. Microbiologists started using Hans Christian Gram's stain, and they determined that it was a very useful stain in microbiology, especially in bacteriology, because it separated the bacteria into two large groups, those that were gram-negative and those that were gram-positive. The gram-negative cells do not retain the first dye, crystal violet, in the staining procedure, and we call them gram-negative cells. The other cells, the gram-positives, do retain the crystal violet, the first stain in the gram procedure, and these we call gram-positive. And why the stain is so important is that this stain separates most bacteria into two large categories, those that are gram-positive, staining purple, and those that are gram-negative. They're not purple, they are pinkish or reddish. Any questions there? You know, there are a few cells that do not stain well, if at all, with the Gram stain. And we'll talk a little bit about them later. The reason why they don't stain well, if at all, is they have a lot of mycolic acid in their cell wall. Mycolic acid is lipid. The Gram stain is a water-soluble stain, and so it does not get into those cells, at least well, and stain them well. Most of the time, it does get into the cells a little bit, and it lightly stains them. But not always, depending on who does the gram stain and how long you stain it for and how good the gram stain is. So oftentimes it will work on these cells, although it stains them lightly instead of darkly. But sometimes it doesn't work on these cells. And it's just that the gram stain is water soluble, and these cells do not allow water-soluble molecules into the cells. Any question about any of that? All right. So let's get started on the gram stain. You need to know the gram staining procedure, how we do the gram stain. And another thing you should know is whenever you write it, it should be with a capital G because it's named after a person, and we always capitalize a person's name. Any question there? So the gram stain should be capitalized, and I always capitalize it. Occasionally I grab a page from somebody else, or somebody updates a page and they won't necessarily capitalize it, but I always capitalize it. And I won't mark you down if you don't, but you should capitalize it just because it's correct grammar. All right, so let's go through the gram stain procedure in brief. You get a bacterial smear on a glass slide. You have to make it. You then air dry it and heat fix it. And we'll talk why in a little bit. You then flood the slide with crystal violet, and you do that flooding for 60 seconds. That's to allow the dye to get in and stain the cells. You then immediately wash with cold water, removing the excess stain. You then get rid of the cold water, you just dump the water off. And then you flood the slide with iodine. Iodine can be a stain, and we could use it as a stain, but in this case we're not using it as a stain. We're using it as a mordant. A mordant is just a chemical that intensifies in this case, the stain that is already there. So a mordant intensifies a molecule that's already there. What iodine does is it complexes with the crystal violet and then makes a larger molecule. So the cell is a little darker stained, and the molecule is a little bigger, and we'll explain why you want a little bigger molecule. You then wash off the iodine and then flood the cell with acetone ethanol. It's called the decolorizing agent, but you only do it for about 5 seconds. What you should do if you were in the lab, I'd show you, is this is your slide and you got cells on there, and what you do is you use a what do you call that, eyedropper, to put on the decolorizing agent, usually acetone and ethanol, and then just let it drip down until you see the color stop dripping. And that will happen in about 5 seconds. What you're doing is you're removing the crystal violet from the gram-negative cells as well as from the background of the slide. But you're not pulling it out of the gram-positive cells. And that's why you need to do it for only 5 seconds. And like I said, the best thing to do is not to time yourself, but actually watch it. And then you just add it until it stops running, the crystal violet stops running, and then you stop. You then need to wash it immediately because the decolorizing agent, even if you flick it off, there will be a little bit on the cells and it will keep acting. So you need to wash immediately to wash the decolorizing agent off the slide. And if you do not, the decolorizing agent will continue working. And it will, if you give it too much time, it will remove the crystal violet from the gram-positive cells. Any question about any of that? Meaning the decolorization step is very tricky. It has to be done for the proper amount of time and not under and not over. If you do it over the time it should be, you're going to start pulling out the crystal violet from the gram-positive cells. If you do it under the time you're supposed to, then you're not going to pull out the crystal violet from the gram-negative cells. And it's supposed to come out of the gram-negative cells. So the decolorizing step is tricky. You then wash with cold water immediately, and then flood, get off the water, and then flood the slide with safranin. Safranin is the counter stain. It will stain any of the cells that are clear and make them show up better by changing them to pink or red. Safranin is the second stain we're using, the counter stain, and it will get in the clear cells and stain them pink or red. You then wash off the safranin just with cold water and blot dry and examine under the microscope. You may do so immediately if there are small droplets of water. That won't interfere with you or the oil if you use oil on the under the specimen or over the specimen, with the specimen. Now when I ask you for the procedure of the gram stain, you should tell me, well, you must start with a bacterial smear. You flood with crystal violet for 60 seconds, flood with iodine for 60 seconds, flood with acetone ethanol decolorizing agent for about 5 seconds, and then flood with safranin for about 1 minute, but it can be up to 2 minutes. And then you should put, and then washing after each step, meaning when I ask you for the procedure, you do not have to tell me after each step you do a water wash. But one place, either at the start or the beginning, you should say after each step you do a water wash. Any question about any of that? Oh, what else did I want to talk about?
I got a question.
Yeah.
Well, you think that that we can use betadine, but I mean, I find it that it's an antiseptic. When you say,
Okay, I'm going to turn up my speaker. Can you say that again?
all right. I work in the medical field, and for me, when you mention that you use betadine with a dropper on the flat, to me, betadine is an antiseptic, so.
I didn't call it betadine. You're using, usually in the decolorizing agent, you're using acetone and ethanol mixture.
No, I mean iodine. I'm sorry. That's what I meant.
Oh, iodine. Sorry. Yeah, you use grams iodine as a mordant in this step. It is used, iodine, although I don't know which form of it, probably betadine, is used in the clinic as a antiseptic.
Right.
Okay? And you can use it as home as an antiseptic. And it's used you can use it to stain starch in cells, like plant cells.
Interesting.
So iodine can be used as a stain. And in this case, we're not using it as a stain.
Gotcha. Okay.
Okay? All right. Good question, but betadine is slightly different than the grams iodine. I think grams iodine, it's just regular iodine, in a alcohol. No, it's not tincture of alcohol. Tincture of alcohol iodine. It's just iodine in something, but I think it's in water-based something. Anyways, it's called Graham's iodine. It's a specific mixture of iodine that Graham made for using in this Graham stain. All right, let's go on to the next slide here. So this is going through the Graham staining procedure, looking at the color of the cells in brief. Initially, when you stain the cells with crystal violet, all of the cells take up the crystal violet. So they're all at this stage staining purple. And there could be an exception if you have cells with a lot of mycolic acid in them. They would not take up the crystal violet and they would remain clear. Most of the times they take up just a little bit of crystal violet and they will be lightly stained purple. But we're going to ignore the cells that don't stain well for the Graham stain, at least for the moment, talking about this slide. So we're going to say that all the cells take up the crystal violet. You then add iodine, and it really doesn't change the color of the cells. It is true they're a little darker purple. The color is more intense and slightly more purple. Like this is a blue purple, it will be a more purple color. But it's very slight difference in the color there. That's all the iodine does. And what we're doing is complexing with the crystal violet, making it a larger molecule. You need the larger molecule because the next step is the decolorization step. You're putting on the usually an acetone alcohol mixture to remove the crystal violet from the gram-negative cells. It does not, as long as it's done appropriately and for the right amount of time, it does not remove the crystal violet iodine complex in the cells if they're gram-positive. Okay? As I stated, if the decolorization isn't done appropriately, meaning if it's done too much, you will remove the crystal violet from the gram-positive cells. And if it's not done long enough, you will not remove all of the crystal violet from the gram-negative cells. But if it's done appropriately, you will have cells, if you have a cell mixture of two cells, gram-negative, which will be staining clear, and the gram-positives, which will be staining purple. So that the decolorization step is the differential step in the Graham stain. We then apply safranin as a counter stain, and it does stain the gram-negative cells pink, and then the gram-positive cells retain the crystal violet iodine complex, and so they are purple in color. Any question about any of that? I had a question to ask you, and now I've lost it. I don't remember what it was. I did mention that this is the differential step and the decolorization step. Now I remember what it is. Why does the safranin not stain the gram-positive cells reddish? Why don't these cells look purple and red? Why do they only look purple? The gram-positive cells. Obviously, the safranin got in the gram-negative cells, and they stained the gram-negative cells red or pinkish here. Why did it not get into the gram-positive cells and stain them a mixture of purple and red, or even, since it's applied second, after the crystal violet, Why didn't it make them stain red? Does anybody have a guess?
I mean, they were already kind of full of the purple, maybe? I don't know.
Yeah, that's actually it. They can, the safranin can get into the cells, and it is there if we were to look for it with an electron microscope, meaning you can see purple color in one region of the electron microscopic image of the cell, and a red molecule, which is the safranin, inside the cell. But when we look at it with our eyes, all we see is the more intense color, which is the purple. So the purple dominates, and we can't see the safranin unless we look for it with an electron microscope. Okay? And when we look for it with an electron microscope, okay, one small region will have the red, and there's no purple there. And then someplace else in the cell, there will be the purple. Okay? So it's just the way our eyes see it. We see the more intense color, which is the purple. The safranin is there, but we don't see it. All right? Any questions about the Gram stain as we've discussed here? Come on. Not getting in there. Oops, wrong one. All right, if there's no questions, this is where I'm going to break. You're getting off early, but we're in good shape. I don't want to start on the next slide because it's going through the Gram stain in procedure, and I'll never finish the Gram stain in the amount of time we have. So we're going to stop early, and I'll see you at 6:30 for the lab.